Hello, welcome to Gene Technology lecture video number 4. This is our final lecture video on this concept. So, the end of this unit, we are going to talk about the different types of technology also that are available. We've already covered restriction enzymes, we've already covered gel electrophoresis and DNA fingerprinting or DNA profiling. We talked about PCR. We've talked about recombinant DNA. Now we're going to get into a few other concepts, but not in as much detail as we did those others. So let's begin. Let's begin by talking about the Human Genome Project. Well, what happened in the Human Genome Project? Well, we mapped the human genome. All of the DNA in a human, all of the chromosomes have been mapped. We know which chromosomes they are attached. We know where the genes are located, on which chromosomes, and where on the chromosomes they are located. For humans, we've figured this out. We've done it for many other species also. It is actually not that difficult to do anymore. We have a company here in San Diego that is involved in that. If you want to go work in that field, biotechnology is a big field. We discovered during the Human Genome Project that most of our DNA, 98% of it, non-coding DNA. 2% of it makes the proteins. That's the coding DNA. We discovered a bunch of little things. We discovered that mRNA can be cut and reconnected. That's called splicing in different ways so that you can actually make different versions of a protein. We also discovered that humans have these things called single nucleotide polymorphisms. What it means is one letter is different in our DNA in some areas, in some particular genes. These particular genes, in a lot of cases, are disease genes. All right. So we can actually use the fact that only one letter is different in a disease gene. We can figure that out, and we can use that information to identify whether someone will or has a particular disease. If you remember, we did this example when we did an example in class. We did a sickle cell laboratory, or when you diagnose diseases using restriction enzymes. If you probably remember back, there was one difference between healthy DNA and sick DNA. Well, one difference means it was a single nucleotide polymorphism or a single nucleotide difference. DNA microarrays. Now, this is a newer technology. I will not lie to you guys. I did not get to do this when I was in college learning biology. This was not around quite yet. With the advancements in technology, I'm not just talking about biotechnology, computer technology, and biotechnology combined. We now have this really awesome tool called the DNA microarray. So, what does a DNA microarray do? Well, it looks kind of like this. It's a board with colors, all right? What do the colors mean? The colors represent whether genes are turned on or off, and you're comparing two different types of cells. We will be doing a DNA microarray simulation in class. One will be available for you to do in class. There's a website that allows you to do it. So what are you going to be doing in our DNA microarray lab simulation? What we're going to be doing is we are going to be comparing cancer cells to healthy cells and seeing how they perform under what's called a microarray. So a microarray basically has each little spot in a microarray has genetic information in it. Each spot is associated with a particular gene. The colors that you see light up are associated with whether it is a healthy gene or whether it is a cancerous gene that is being expressed. Okay. Sometimes both are being expressed, so you will have a medium color showing. All right. So we are comparing for, say, for example, we took a sample of what we think is skin cancer from somebody, and we took a sample of what we believe is healthy skin from that same person. So we will compare the genes in their cancer cells versus the genes that are turned on and off in their healthy cells. So these colors tell us which genes are lit up, meaning they're on, or which genes are, if they're black, it means the genes are totally turned off in both cases in a cancer and a healthy cell. Okay. So what you do is you tag normal cells and cancer cells with different colors. So in this example here, the green is showing you healthy DNA being expressed. The red is showing you cancer cells expressing. The yellow is the in between. That means in those genes are being expressed in a cancer and a healthy cell. Okay, so we use microarrays in many ways now. I'm explaining to you how we use it to tell if a cell is cancerous or not. We can actually compare people to each other. For example, they've used it to look at identical twins. And even though identical twins have the same exact coding in their DNA, some of their genes are on while some of them are off. And those are variations in twins. So if you look at identical twins with the same exact coding, their microarrays will look different. They will not necessarily have the same genes turned on and off. So we can compare twins to each other and we can overlap them and look at which twin has genes turned on and which twin has genes turned off. Basically, like I just said, they're two-dimensional visual arrangement. You're using computer chips, you're using a computer to do this, and you will see how this all works when we do our simulation in class. Gene therapy. This is another method of gene technology. What have we discovered? We have discovered that we can incorporate DNA into cells by using a virus as our vector, so as our spaceship that transfers the foreign DNA. Viruses, as a little bit of background information, they generally contain DNA or RNA that enters the cell. The DNA and the RNA now cause the cell to behave differently. Usually, these viruses will infect a cell, have the cell produce more of the virus, and then a lot of times will kill the cell after they've produced more of themselves. So viruses generally, they infect and they a lot of times lead to death of that particular cell. And after the death occurs, they spread more of their virus to more cells and they continue the process. So what we have discovered is we have figured out ways of genetically modifying viruses to carry, in this case, this picture is showing you a protective gene is being inserted into the genome of an individual. That protective gene is being sent in via a virus that now enters. So the virus is like the spaceship that gets it into the cell, and then as it infects it now with a protective gene, and now the actual cell has been changed. So we have now infected the cell with healthy DNA, protective DNA, so that the cell now has been modified. So we've done what's called gene therapy. We've modified the bad genes with possibly new genes. Okay. The easiest way we've discovered is to do it with a vector. And I already told you that's like the spaceship that transports it. And viruses are commonly used as the vector to transport it. Okay. Now, a clone, if you hear the term a clone is an exact genetic duplicate. It could be a duplicate of a piece of a DNA. It doesn't even have to be a whole section of DNA, a piece of DNA. It could be a whole cell. It could be a complete organism. Okay. We discovered that you can actually clone animals because everyone thinks, like everyone talks about cloning a human. Can you clone a human? Scientists have the technology currently, yes, to clone humans. Are they able to do it legally? No. You will get in a great deal of trouble if you do testing on humans, especially something of this nature where you're going to clone a human and then that human could potentially be born with a lot of health risks, health problems, and they're still a human and they have rights as a human. So cloning humans is not allowed yet, and I don't know if it will be allowed in our lifetimes. But what they did is they took the sheep named Dolly, and what they did is they added DNA to a donor egg. So let's talk about this whole process. So it's called cloning by nuclear transfer. All right. So what you do is the introduction of a nucleus from a body cell. All right. So let's say they take a cell from a sheep. Okay. They have a donor cell from a body cell or a somatic cell. So you get a donor cell. So now you have a donor nucleus. So why is it a donor nucleus? Because it contains all of the chromosome to produce the sheep. Okay. Then it is inserted into a donor egg cell. But before they take that donor nucleus and put it into an egg cell, they have to remove the old DNA that was in that egg cell from the egg that was given up by another one. Okay, so you take an egg, you remove the DNA from it. So the nucleus of the egg cell is removed. In that egg cell now that doesn't have it doesn't have any DNA in it, you now import or add all of the DNA that came from a body cell from another sheep. So now you have an egg that has all of the exact DNA from the original donor over here. Okay. You take that egg, all right, and it gets imported back into mama. So they put it back into mama. And while it's into mama, this egg now is an embryo and it starts to divide and it starts to make more copies of itself. Okay. And then mama gives birth now to a clone of the original sheep. All right. So it all starts with putting donor nucleus into an egg. The egg starts dividing. It gets put into uterus, the baby, and then it is now the mom gives birth to a clone of the original sheep. Now, when they did this with the sheep, they called her name was Dolly. It happened in 1996. Dolly died about halfway through a typical lifespan. Okay. Why did that happen? So sheep live about 12 years apparently, and Dolly died prematurely at the age of 6. And it pretty much died. Dolly died of old age at the age of 6. So what we discovered is that Dolly was produced with donor DNA that was already older. It was about 6 years old already. So the donor DNA was about 6 years old. So the Dolly, the sheep, only lived an additional 6 years because sheep only live about 12 years approximately. What did we discover? We discovered that the DNA was old. We started with old donor DNA. How do we know the age of DNA? Well, we know the age of DNA by these things called their telomeres. The end of the DNA is the telomeres. You got to think of them kind of as a protective cover. All right. What do? They protect DNA from deterioration or from sticking, fusing with neighboring chromosomes. So they're protection of our chromosomes. As cells divide and divide and divide and divide and divide, the ends of the DNA called the telomeres, they shorten over time. They degrade over time. It is believed that the shortening of our telomeres over time is what leads to aging of an individual. So they believe that Dolly, when she was cloned, Dolly was cloned from an older sheep, and that older sheep's telomeres were shorter. Therefore, Dolly's lifespan was shortened as a result of beginning with a shortened life. For example, you began with shorter telomeres, you didn't live as long. So this is where the issue comes into play. Like, let's just say, okay, you want to clone a human. If you clone a human like myself, who is already older, that means the lifespan of my clone will not be very long because I am already an older individual. So if you use 40-year-old DNA, they will be born a child with 40-year-old DNA. They will start developing ailments, diseases that normally you would have as an older person, but they would start developing at a younger age. So there's an inherent problem with cloning a person. Now, some species do not experience telomere shortening. Inherently, basically, they are almost, you could say, almost immortal if their telomeres don't shorten. They have a mechanism, special enzymes that rebuild back their telomeres after they divide. So their telomeres completely get rebuilt back. So their DNA, in essence, doesn't age. Okay, so some species don't experience it. Now, vaccines. These have been a hot topic lately, especially because we've been dealing with something called COVID lately. What is a vaccine? Now, this isn't red because I've added it to our notes. It wasn't before. This is a new thing now because in the past, a lot of us never had an mRNA vaccine. We had a typical vaccine was you got injected with harmless version of a pathogen. A pathogen is a disease-causing agent. So you got injected with a harmless version of a disease-causing agent, and your body had what's called an immune response. I'll explain to you what an immune response is in a second. That immune response prevented you from getting sick from it when it showed up again. Well, what we have now is we have old vaccines with a harmless version of a pathogen. We have new vaccines now that use mRNA. Let's do a quick review. mRNA is what DNA gets rewritten as during transcription, and then the mRNA gets translated into a protein. All right. These new mRNA vaccines, what they do is you get hit with an mRNA vaccine, you get injected with it. The vaccine then puts mRNA into your cell, Your cell takes that messenger RNA and produces a protein. Okay, so what are they doing right now with, for example, like coronavirus? We are getting injected with if you get the vaccine, you're getting injected with an mRNA vaccine for I believe two of the different types of vaccines that are out for it. Two of them inject you with mRNA. You produce what's called the spike protein, which is on the surface of, for example, COVID-19. You produce a spike protein that is on COVID. That spike protein then your body can now have an immune response to that spike protein. So rather than getting infected by a full-blown virus that can now make you sick, you are going to have an immune response to just the spike protein. Once you have an immune response, you can then defend yourself against the illness or the virus when it shows up again. This technology, by the way, is also being used currently in cancer treatment. They are creating an mRNA sequence using a protein found on the cancer cells. Your body then makes a protein that is on your actual cancer cells, and then your body has an immune response to that protein and then begins attacking your own cancer cells. It's kind of the future of cancer treatment, and it's pretty awesome, actually. Okay, it uses similar technology as the mRNA vaccine. So what does it mean when you have an immune response? When you have an immune response, it means your immune system, your white blood cells produce antibodies. That means you have had an immune response. You produce antibodies in response to something foreign. The foreign substance that is infecting you is called the antigen. So, for example, in this case, so you make antibodies in response to an antigen. So a typical vaccine, we inject you with a harmless version of the pathogen. Okay. That pathogen is an antigen. It is a foreign molecule. And what did it do? It stimulated antibodies. You made antibodies, which means you had an immune response. So the whole idea of a vaccine is to create an immune response that allows you to make antibodies. What do antibodies do? Well, one thing they do, they stick to the antigen. So when the bad thing, the pathogen shows up to make you sick, antibodies stick to it. They label it. Antibodies in some cases can actually kill the antigen. If they cannot kill it, they label it and they inform your other white blood cells that can come along and actually do the damage and kill it. So antibodies are labels of bad guys. We have memory. So when you get a vaccine, your body has memory cells in your immune system, and you are able to make those antibodies very quickly so that if a bad pathogen shows up, you are now immune to it because your antibodies will stick to it immediately and kill it or remove it before it actually gives you symptoms or makes you ill. Finally, gene technology has led us to these things called GMOs. If you ever see the term GMO, we are talking about a genetically modified organism. A lot of people refer to them in the food world, okay, because they're used a lot. GMOs are used a lot in the food world. This right here is a genetically modified frog. This frog was genetically modified to have transparent skin. Why would we want a frog with transparent skin? Well, these frogs are used in research in science classrooms. And what are they used for? So that people do not have to actually dissect. They can actually see right through the skin and see the working parts of the frog and how they are beating, the heart beating, for example, or how things are moving along and what is happening internally without actually having to cut open the frog. So they are used in research. What have we done to foods? Well, we have genetically modified a lot of foods to make their own pesticides, actually. A lot of foods are created so that they can produce a pesticide so that insects don't like them, and they make their own chemical pesticides. A lot of them are genetically modified to be resistant to weed controlling chemicals. So why would we want to make something resistant to weed? So if I'm farming, I can use a lot of pesticides to control weeds, herbicides they're called, and then I will kill all the plants except the plants that I am desiring to grow. So that is a common thing that is done with corn, soy, a lot of different fruits, and you'll see a list here of all the different ways we have genetically modified. We've also genetically modified some, for example, rice. We've genetically modified rice to produce vitamin A. Okay, so you can modify it to produce things like vitamins. You can modify it to be bigger so that you can have a bigger yield. They are now modifying salmon. Salmon is now available. I believe they've genetically modified salmon. They've combined three, I believe, different types of salmon, and they've produced a hybrid of the three. They took one that reproduces quickly, one that grows quickly, and one that has larger meat, produces more meat. So they've done this so that for food production primarily. So you can they've genetically modified some crops to be more resistant to colder temperatures. So you can do quite a few things with genetically modified foods. All right. And the whole goal is to feed more people. That will be the end of lecture video number 4.